So I'm going to start out with the way I always start out in these cases, which is thinking about a patient. And this is actually, unfortunately, a patient of mine that, as you'll see, I never got to meet. But she was a 6-year-old girl who had been previously very healthy. Over the summer one day, she was playing in the pool, and her cousin, thought it was funny, so pushed her into the pool. And when she did, she actually didn't come up right away. And luckily, her dad was there, saw her, pulled her immediately out of the pool when she wasn't coming up for air, and saw her basically roll her eyes to the back of the head and, was unresponsive briefly. Luckily, she responded relatively quickly. They didn't have to do anything heroic. She just kind of came to after a few seconds. But he took her to the emergency room because he was clearly shaken up by this. When she went to the emergency room, they basically gave her a clean bill of health. So they did an exam. She had a normal exam. They did an EKG. They looked at all of the electrical impulses, measuring even the QT interval, and all of that was fine. They even did an EEG, so they looked to see if that could have been a seizure. That was fine. They checked immediately her glucose when she came in. It wasn't hypoglycemia. And so people didn't know what was wrong, but they thought they had done really looking for any of the most common things, and so said, basically go home. We don't see anything wrong here. We don't know what happened, but we don't have an etiology. So she went home, and then, again, this was during the summertime. She was away at summer camp, at the YMCA summer camp. And as they were doing that, of course, many kids learn to swim in summer camp. So she was there learning how to swim. And she was in the pool, and the lifeguard was dutifully watching her. And when she was there swimming, something similar happened where she slipped under the water. And when she slipped out of the water, again, the lifeguard was right there, recognized what was going on, pulled her out. But in this particular case, she didn't come to right like that, like when her father was with her. So the lifeguard was CPR trained, immediately started CPR. They didn't actually have their defibrillator, but had started CPR. They called 911. It took EMS about 12 minutes to get there. By the time they got there, they shocked her. She was having an arrhythmia, so they shocked her, intubated her, and took her to the emergency room. When she got to the emergency room, again, they did obviously a big evaluation to try and figure out what was going on. She had an EKG that looked okay. She had an echocardiogram. Again, those of you who are clinicians will understand this. Her heart wasn't pumping all that well at the time, but she had also been down for 12 minutes, so that was somewhat understandable. They imaged her brain, and her brain didn't have any, for instance, bleed or hemorrhage. There was clear evidence of hypoxic damage because she'd been down for 12 minutes, but they didn't see any specific other problem. During that time, again, she was intubated, and as they were evaluating in terms of her brain function and her brain activity, ultimately the neurologist had the conversation with her parents that, in fact, she was going to be brain dead after this whole process, or at least she would not be back to a functional status, and her parents made the decision to withdraw life support based on that. But they were quite concerned about what could have caused this. The doctors were concerned. They were concerned. There was no family history of anything like this that had ever happened. It was quite shocking. But importantly, they had another little girl at home, and they were very worried about the chance that this could have been something potentially hereditary and could it happen to someone else in the family. And so at the time, they had a cardiologist who did a great job. This was actually a couple years ago. Took a blood sample, sent it off essentially for a panel of genes associated with causes of sudden cardiac death or sudden cardiac arrhythmias, and sent it off and did a sort of, kitchen sink approach, tested for everything they could, took a requisition and basically check anything that was a possibility. In some ways, understandably, because this was going to be the last chance. She was unfortunately not going to be able to survive to be able to give us any more hints about what went on. So as this went on with the genetic testing, and just for those of you who don't know, the genetic test oftentimes takes something like 8 weeks to get results back. So unfortunately, you can understand the family's process with this. They bury their daughter. I mean, they're going through the grieving process, and then they get back these genetic test results, which then it comes back all over again. And again, she had testing. These acronyms probably don't mean much to many of you, but testing for a panel of genes associated with long QT syndrome, a panel of genes associated with arrhythmogenic right ventricular cardiomyopathy, and then another panel of genes associated with catecholaminergic polymorphic ventricular tachycardia. And all of that gobbledygook, for those of you who aren't clinicians, those are really just different causes of arrhythmias that can result in sudden death, and that was what they were looking for. So again, throwing the kitchen sink at this, perhaps not surprisingly, and we'll get into exactly what you do when you do this type of testing, the test report came back that there were actually 3 different genetic variants that the laboratory said, we're not sure. We're not sure if this is actually the cause of whatever happened, but we see something in a gene called CAV3, which is associated with long QT9. We see a change in a sodium channel, which is associated with long QT3 or Brugada syndrome. And yet, that's not enough. We see a 3rd change in the ryanodine receptor that's associated with ARVC, as well as catecholaminergic polymorphic ventricular tachycardia. And so the point that I want to raise in doing this is the family, in many ways in an understandable way, having gone through losing their child and being concerned about their other daughter, is now left with a bunch of gobbledygook to them without clarity in terms of which one of these is actually pathogenic and the cause of their daughter's death, potentially. Is any of these the cause of their daughter's death, or is there something else that's going on? And importantly, by figuring out what it is, being able to help other family members. And so we're going to go through just how do you actually get to the point of then interpreting that information, what can you do in those types of clinical situations to be able to help the patient and help the family. So I apologize for some of you that this is going to be feel like high school biology, but I just want to make sure everyone in the course is on the same page as we're talking through these things. So when you think about the genetic information that we've got to think about, for some of us it might feel daunting at first pass, but in many ways it's getting much more understandable on a day-to-day basis. So we're talking about 3 billion base pairs, 20,000 genes approximately at latest counts. And when we think about how these are organized, importantly, there's a lot of the genome that we don't understand, that it's organized in this way that looks almost like gobbledygook until we show you or bring out that there are certain parts of the genome that encode what we think of as the proteins or the gene products that come out of this, and it becomes obvious that there are little bits of golden nuggets that are embedded within the genome, and that we have to figure out ways of putting that together to making sense. So again, just as a vocabulary way of doing this, we call those golden nuggets the exons over here, and many of you have heard me use the term exome, which is just the aggregate of across an entire genome, all of those exons put together. But yet there are other parts, and this is one of the main points I want you to get from today, there are other parts besides the exons that indubitably hold important information, and in some ways we just don't realize what we're missing with all of that information. So in particular, there are regulatory regions, so there are ones that are quite proximal to the genes that we call promoters. But in addition to that, there are things that affect splicing. Splicing is the process of cutting and pasting and putting together these different exons, and I'll explain a little bit more about that in a second. There are parts of regulatory regions that are within the spaces here, these intragenic regions. There are also things that affect splicing, splice enhancers that are embedded within these, and I won't have time to go over many of those. And then there are things that we call locus control regions that actually affect gene expression for a large number of genes that are physically either clustered together, either linearly on a piece of DNA or in a 3-dimensional conformation when those genes physically come close to each other in terms of touching each other. Even though they might not be close on a chromosome, physically they're close within the nucleus of a cell. And so with all of that, these all can affect actually the amount of a gene that's expressed or where it's expressed or when it's expressed, and therefore be involved in terms of human disease. So one of the important signals that the cells are getting in terms of being able to put all of this information together is very precisely understanding where do you cut and where do you paste. And with this, what I'm meaning to show here is that there are some sequences that are very important to signal when that happens. The size of the letters here tells you how invariant those positions are for the correct cutting and pasting. So for instance, at the 5' splice site, you absolutely have to have a G here, you absolutely have to have a T here. If you violate that, you don't get the correct splicing. On the other hand, what you can see is that in any of the other positions here, there's a little bit of flexibility. There may be a preferred nucleotide, but there's more flexibility. And so the point that, or the impression I want you to get is that in general, there aren't hard and fast rules about this. And so when you try and just look at a sequence and make a prediction about whether or not it's going to affect splicing, it's just a prediction. I mean, that's the point that I want to make, is that it's not based on hard experimental data. They're predictions that we're making. In addition, this is what I was alluding to, in the middle of these introns are actually branch sites that have conserved sequences. Just so that you know, we oftentimes don't interrogate these regions here. And so we actually don't know what we're missing when we think about those splice junction sites or in fact many of the regulatory sequences. In addition, several of you probably know that a new field, at least new in the past 5 years, are microRNAs. These microRNAs that are actually affecting expression of genes, in fact, many, many different genes. I won't go into lots of detail about that, but many of those are embedded within the genes themselves, either in the non-coding regions or in some cases the coding regions. And again, it's an important thing to pay attention to if you're thinking about gene regulation as being a potential risk factor for disease. Within this, in terms of the actual, how this looks, as I said, there are these regions here in terms of cat and cataboxes, which are, again, conserved regions that tell the protein translational expression machinery where to start actually being able to make the messenger RNA, and again, conserve sequences that we oftentimes interrogate because mutations in those regions can actually be problematic. Beyond that, though, one of the things that very, very few of us, to be honest, experimentally look at is actually the three-dimensional structure of the chromosomes themselves. We oftentimes are reading out linear information, but we're not thinking about how this actually functions in this way. And I was starting to describe this to you, but I happen to know a region on 16p, for instance, where there are 2 copy number variants that are in different parts of 16p, but if you physically look at how they are, even though they're far apart on the chromosome, physically, when they're in the cell, they actually come into close proximity. And in fact, they have a very, very similar phenotype, in large part because they may actually be interacting within the cell. And so it's just one example of how this happens. The other thing I'll say is that much of this in terms of the chromatin modification, or these different proteins that are involved in terms of sort of wrapping the chromosome material around and consolidating, making the structure of the chromosome, many of these genes we now know to be involved in several developmental processes. So things like congenital heart disease for sure, autism spectrum disorder, other neurodevelopmental disorders is a common theme that we're seeing because oftentimes these types of genes have profound effect in terms of gene regulation in a very global sense. So that hopefully helps in terms of that. For some of you, the graduate students in particular, this won't come as new news, but there's a huge project that's been funded by NHGRI to specifically now look at the non-coding regions of the genome. So with the idea being that there's a lot that we've been missing, and so the ENCODE project has been literally big science, big data, big science to be able to collect information about those non-coding regions. I have to admit I'm not an expert within these regions, but the point was to use all sorts of different essentially technologies and platforms to be able to characterize all sorts of things like, for instance, what parts of the genome are methylated, what parts are actually open in structure and sort of available for transcription factors to be able to bind, what different parts of chromosomes are actually touching each other, being able to look at expression profiles across different tissues, looking at epigenetic changes, and essentially cataloging and putting all of that information together within browsers where you could actually consolidate that and think about that in terms of any one gene. And I won't go into lots of detail about this, but this involves RNA-seq, ChIP-seq, transcriptional expression, and even down at this point to being able to do this on single cells, so one can actually do this even within a heterogeneous organ, looking at different cells within that organ and developmentally looking at different time points. And at this point, for anyone who's interested in doing it, this information has now been cataloged and put together, for instance, in genome browsers, so that you can look at a linear piece of DNA in this particular case, and with each one of these tracks be able to look and see, for instance, are there areas that seem to be very highly regulated in terms of transcription? Are they open domains? Are they methylated and epigenetically modified? And so you can map out regions like this region here, for instance, that seem to be more active, if you will, and one would predict that those regions might be places where if you started, futzing with that, maybe it wouldn't be such a good idea. And so, in fact, one of the things people have wondered about is, if you think about the disease burden, where is the disease burden? And it's interesting that it looks like the common theme that we're seeing is that for rare diseases, the common burden is basically within the coding sequence, as opposed to when you're thinking about common diseases, it may be the case that much more of that is within this non-coding region. And so depending on whether you're interested in sort of garden variety type 2 diabetes versus, perhaps a very, very severe childhood disorder, the genomic architecture may be quite different in terms of that. So as people have thought about that, this is one particular case, in fact, with inflammatory bowel disease, where folks had done GWAS studies to try and understand where the signal might be coming from, and the signal was mapped to 5p13. But interestingly enough, there wasn't a gene that was exactly within the region where the SNPs seemed to be piling up, and you seem to see the signal. And when you start putting the ENCODE data around in terms of this, it actually maps within a region that's essentially a gene desert, places that don't have much in terms of coding sequence, but it piles up in the region where, again, you'll see this pile up between all different types of looking at the non-coding regions, where transcription factors are binding, where the DNase is able to get in to the chromosome. And it looks like in this particular case, there's probably something there that's actually regulating the expression of the genes in that case. And so it's, like I said, just an interesting tool, especially for those of you who are interested in common diseases and particularly perhaps common variations as a susceptibility to common diseases. Just let you know there's just an army of data that's behind you in terms of being able to use that information, catalog it, put it together, an amazingly underutilized resource in my opinion. So within that, the reason I'm bringing this up in particular is that one of the arguments you may have heard from some of us is, you know, as we're doing this, do we need to sequence, in fact, an entire genome? You know, how much do you get from an exome and how much additional data do you get from a genome? And do we need to think about sequencing an entire genome to be able to get what we need from this? And so, you know, for those of you who have been listening to precision medicine and hopefully this new initiative that's going to be funded, the Million Americans study, the idea behind that is it's going to be genome sequencing of 1 million Americans that, you know, as the price is coming down to perhaps about, you know, $1,000 per genome to be able to get all that information, because so much of, in fact, from a public health point of view, the disease burden is really within those non-coding regions that we need to make sure all of that gets covered. Just so that you've heard all the arguments, again, when you think about the exome, so whole exome sequencing versus whole genome sequencing, again, I think it depends for the disease on what you're interested in studying. I think for rare variants, especially ones that are extremely impactful and affect reproductive fitness, you're probably getting a huge amount of information from an exome alone. On the other hand, those of you who are interested, again, in studying more common diseases that don't kill you by the time you get to be 20 or 25, it's more likely that the genome is going to reveal more information. And if an exome doesn't give you the answer, it's possible that a genome will give it to you for all sorts of reasons. Either it's a structural variation you can't see within an exome. You may get, just for other reasons I could go into from a technical point of view, you may get more even coverage or be able to see insertions or deletions within genome sequencing that you might not see on exome sequencing. So this is a debate that's currently going on for us. This has actually been studied. The number of studies is relatively small at this point, but some of you may have heard folks like Bob Darnell talk about this. This was a study that came out this summer that's comparing the diagnostic yield for intellectual disability between different platforms. So for instance, if one started with a chromosome microarray, not bad. You can get 12% of cases figured out that way. If you then go on to exome sequencing, you can get another about 1/4 of cases identified by that. But the argument, at least from these authors, is that if you continue and go on to genome sequencing, you can probably pick up another 42% of cases that way. And so again, the argument being that if you keep, especially for genetically heterogeneous conditions, if you keep pushing and get more and more data, you'll explain more and more of these cases. So interestingly, if you were to aggregate all of this, if this actually scaled and were true not just with 100 cases, but now with thousands of cases, this would suggest that the burden of intellectual disability is mostly genetic, in terms of that being, at least 80% that's already explainable by these different technologies. So anyway, there are other studies that are saying similar things. In this particular case, there's the exomes could have been done a little bit better using modern technology, so I think the yield is probably even higher than what's reported here, but it does definitely show that there is something to be gained from genomes over exomes. So again, I just want to review some very, very basic concepts because it'll be important as you think about what the other things we're trying to teach you. As this information gets processed, these are the nucleotides in sets of 3. In other words, a codon will actually encode the different amino acids. And so we talk about this as a reading frame. So in other words, this set of three different units at a time. And if you were to knock that off by taking out or adding, for instance, one single nucleotide, it would result in what we call a frame shift or changing that reading frame. And that's generally not a good thing. This is the Triplet Code, which you can look up anytime you need to, but it's the translation essentially of those nucleotides into the amino acids. And within this, one of the things that you should always just realize is that our genomes are not stable. And so within this, there's the ability to actually pop genes in, pop genes out, pop genomic regions in, pop genomic regions out. And so one of the things that we think about is not just the sequence itself, but actually looking at the copy number or the numbers of these copies. And if one simply reads out the digital sequence itself, it can sometimes be quite difficult to know how many copies you're looking at. If you're looking at 1, 2, 3, 4 copies of a gene, perhaps, it just looks all potentially like normal sequence, although the computational methodologies to quantify this are certainly getting better as time is going on. One of the challenges from a technical point of view is that if you look at our genomes, all of our genes are not necessarily unique. There are definitely gene families that have arisen from an evolutionary point of view through duplication events, gene duplication events, and then divergence. And so from a technical point of view that many of you will not want to get into, we sometimes have difficulty resolving one gene in a gene family from another gene within a gene family. And so again, just sort of pointing out some of the weaknesses of some of the technologies, I just point this out because some of these, for instance, can be important genes, for instance, with muscular problems, myopathy problems, immunological problems. So just realize that almost no genetic test is ever perfect. As this happens, this is just an example of what's happened evolutionarily in your globin genes, and just to realize that, again, there have been these gene duplication events, which actually in a good way have allowed us to develop fetal hemoglobin as well as adult beta hemoglobins. And sometimes we even co-opt this for people, for instance, who have sickle cell disease by trying to go back and use their fetal hemoglobin to essentially replace or compensate for their adult hemoglobin that has a mutation. Just fast forward through that. The other thing about this is just to remember Barbara McClintock's jumping genes, that our genes are still dynamic, and so they're definitely transposable elements that hop in the middle of genes and unfortunately disrupt them, so that we still see significant evidence of that as a continued form of mutations that we see within patient populations. So within this, there are also sequences that tend to be repetitive or seen multiple, even hundreds of thousands of times throughout the genome. These also tend to be regions that can be unstable, and as a means of causing mutations, are actually not an infrequent cause of the mutations that we see. But on the other hand, most of what you're going to see when we talk about sequencing-based tests are going to be the following. And so I'm going to focus just a tiny little bit on that so that you'll know what we're talking about as we start talking about different methods of testing as well as the implications. So point mutations is kind of a loose term that we often use to say either a single base pair or a small number of base pairs that's affected. We use the term silent when it actually doesn't change the amino acid that's encoded. So, effectively, even though you've substituted a nucleotide, it hasn't changed the protein structure. That may not always be the case because sometimes these silent changes will change things like splicing or splice enhancers, but in general, we, I have to say, sometimes miss those. There will be different changes that cause a premature termination of the protein, and so we often call those nonsense because what happens distal to that point is actually gobbledygook in terms of what was originally encoded by the protein. And then missense we tend to use, or we don't tend to, we use for the designation of one amino acid that gets substituted for another based on that nucleotide change. This actually is the bane of our existence, this idea of the missense change, because it's not a priori easy to just see by looking at it that it's clearly going to be a deleterious type of variant. It may actually be really terrible, but it may not be so bad, and we'll talk about that a little bit more. Otherwise, things like insertions and deletions can be problematic. It depends a little bit on the size, whether it disrupts a gene or not, whether or not that's a gene that's dose-dependent. And as I said, when it comes to splice sites, we have ways of predicting that, but they're imperfect. And regulatory sequences, to be honest, we oftentimes don't even look at, so we don't know what we're seeing. And then for many of the neurologists, they're very familiar with the expanded repeats, which just as a word of amplification, which Jill Goldman always reminds me to make the point of, when it comes to something like exome sequencing, you're not going to be seeing those types of repeats coming out on your sequencing, at least not currently the way those are set up. So just know what you're not seeing. So as we think about this, I want to make a distinction as we talk about the different genetic tests that patients might get between those that are essentially what we would call genotype-based. So in other words, there's a genetic change, I know what it is, and I want to go in and say, basically, you either have it or you don't. Give me a plus-minus readout in terms of that. Those assays are actually relatively easy to set up, run, and interpret, and we love those types of things because they give us clear-cut answers. On the other hand, many things are now sequence-based in terms of giving us this digital readout of all those A's, T's, C's, and G's, which come with the necessity of being able to interpret that information, and that can become somewhat more difficult. If you know what you're looking for, it's cost-effective to do a genotype-based assay. That is, just go in and target in a targeted way, go what you're asking is there. But on the other hand, many times we don't know what we're looking for, and so we end up going on kind of this fishing expedition as we're doing it. When we go on the fishing expedition and we go in terms of looking either on a genome-wide basis or looking at a number of genes, you can come up with several different results, and I've actually simplified this. The new guidelines that are coming out from the college, the American College of Medical Genetics on this, gives 5 different categories. And so there's one category which is basically clearly this is a mutation, and by that I mean it's something that's a disease associated, and at the other extreme, something that's clearly just a normal variation, don't worry about it. And then there's basically all these shades of gray in categories 2, 3, and 4. And for any of you that are aficionados, you can certainly come, and we end up having these sort of bake-offs, where we'll give out 10 variants, we'll have 10 different people interpret what they mean, and it's quite interesting to see that even though these are all CLIA clinical certified laboratories, they don't all come out with the same interpretation, which is a little bit scary, but something that's still a work in progress. The one that's difficult, or the most difficult of these, are the ones that are square in the middle of something we call clinical or variants of unknown or uncertain clinical significance. And oftentimes those are variants that may be seen for the very first time or a very, very small number of times. They're clearly not normal genetic variations, so we don't see them in 1% of the population. We may have never ever seen it before, and we may have only seen it 1 in 60,000 individuals. But those are things that, just based on looking at the either published literature that's available, if it is available, or looking at computational predictions, it's not clear what that information means. And if we go back to that first little girl I was telling you about, that was what was so troubling, is that we ended up actually with a lot of these variants of uncertain significance, and you left being in this very much this gray zone about what do you do with that? Do you, go with worst case scenario that it's really disease causing and take the maximum intervention for that, or do you just slough it off and don't worry about it? Because I will tell you, statistically, 90% of these variants of uncertain significance eventually will end up being downgraded, is my expectation, to being just rare but normal variations that won't be disease causing. But on the other hand, how do you figure out which one of those 10% actually are going to be potentially very important? So one of the things, and I'm just going to throw this out specifically to those of you who are informaticians, is this is very dynamic. And so this is the thing that I am most interested in being able to figure out a solution for. So you guys are smart, and I want you to figure out how to do this. This is evolving, and so what I mean by evolving is that our collective knowledge about genetic variation is clearly evolving, both in terms of patients that actually have clear deleterious mutations, but also in terms of normal individuals. So for instance, when we get to the 1 million Americans, the wonderful thing is that we'll have 1 million Americans sequenced. We'll know what diseases they have and don't have, and we'll be able to have a rich database in terms of what normal variation is. So when that's available, I could go back and reinterpret many of those VUs, like with this little girl, at some time with that. But I'm going to have you help me think through this process. So now, for those of you who are clinicians, think through this. You see a patient, you order a genetic test, you get back one of these variants of uncertain significance, and for your patient, if they're anything like my patients, probably 70% of them I'll be able to find in a year. A bunch of them will have moved on, or will have decided that they are no longer part of our healthcare system here. But over time, I will have fewer and fewer of those patients that I know how to recontact. This will get better as we have at my NYP or other health portals to be able to have connectivity with them. But right now, I'll lose a fair number of them over time. On the other hand, if these variants get reclassified, how is a patient to know that it gets reclassified and be able to use that information in an effective way, right? Because 90% of the time, these things are essentially going to go away and not be concerning. But on that 10% of the time, this could be potentially life-saving for their family or life-altering in terms of that. And so I'm just going to throw out a question. You don't have to vote, but you can vote certainly if you want to. So the other part of this is though if a laboratory, for instance, were to have some way of reinterpreting this information iteratively over time and pushing that out to the clinician, then the clinician now has a responsibility in a certain sense of being able to get that back to the patient, right? Because we're the gatekeepers of this medical information. And so as the clinician, that's now putting also an additional burden on you, right? It's also offering a great way of you to help improve, the care of your patient and to be able to do your job better. But on the other hand, if you've lost contact with some of those patients, that may make you feel really rotten if you're trying really hard and you can't find them and get them this information. So I'm just curious, how many of you, if you were a physician, and well, let me start with if you're a physician or a provider in this situation, would want that type of information back? You'd say, if you set up a system where you could iteratively interpret this information, give me that information back over time, maybe 5 years later even, would say, yeah, I'd sign up for that program. I'd like to have that type of information. And how many of you would say, I don't think so? We'll just skip that one. Okay? Very interesting. Those of you who don't know, Dr. Wapner here is an obstetrician, and so there may be differences in terms of the prenatal to the postnatal side. Okay, now think of yourself as a patient. Okay, so now this is your information. How many of you as patients would say, I would want to make sure my doctor got that reinterpretation over time, that information? And how many of you would say, yeah, let's nix that? It's interesting. Okay. So the next question becomes, and this is what I'm interested in, those folks of you who are informaticians, so how do you effectively do this? I mean, what's the most effective way? I mean, just from the little informal poll that we've done here, right, it looks like at least there's some interest in being able to set up some sort of system to do this. The arguments that I've heard on the laboratory side is that, oh, those doctors, they don't want that information because that's a burden to them to be able to have to, then find those patients, and, is there any sort of legal liability if they get that information and don't find that patient, and, maybe that patient dies but could have been saved by this information, what's sort of, they don't necessarily want to take that on. But on the other hand, as many of you showed by raising your hands, it really could be a good thing. And of course, the hope is that you'll have an ongoing relationship with those individuals. So, I don't know, I'm gonna throw it out to you guys. How do we do that? I mean, how do we set up this sort of learning healthcare system where we can actually evolve in this? The information, I can tell you, this information is evolving very rapidly, right? So it's not that this is not going to happen. It happens every single day. So how do we set this up? Go ahead.
Well, you give the patient the information you have at the time you have it, and then they have to be the keepers of the information, not the physicians necessarily, because that gets lost in a giant shuffle. And then you put everything up online, and then with the caveat that they should then contact their physician, either the original physician or the physician that's taking care of them at the time.
Okay, so I'm going to repeat mainly for the people who might watch the video later. So the idea is empower the patient, if I can summarize, right? So the patient is the one who has the vested interest in this. Give them the power and the responsibility to be able to keep up with this, right? So you could give them exactly their genetic variation, and you could set up something like a web-based portal where they could log in and they could see for themselves, is the classification for this variant changing over time? And so you put part of the responsibility, the shared responsibility, on them. How many of you like that idea? Okay, how many of you say, I don't know, I don't trust those patients with that information, they'll misinterpret it, they'll do something crazy with it? Okay, a couple of people say, I don't know, they may need some help even though you've told them to come back to their providers, maybe they won't do that and they'll do something crazy with the information. Go ahead.
There's some sort of free resource that they can reach out to. So
Okay.
if they lost their health insurance or something like that, they can definitely have a reasonable person to keep that data.
Okay, so the question, is that also, unfortunately, healthcare coverage is dynamic, and some people might lose insurance, and so they might be sitting on this information. They might want to access resources, but not really be able to from a financial point of view. So could we somehow set up a system to make sure we catch those people, right? They want to do the right thing, but they just feel constrained because of resources. And certainly if we do this, we want to be equitable, right, in terms of thinking about how to make sure we don't lose anyone in this process. Okay, so let's pretend that we could somehow solve that problem. It wasn't a matter of access, but I completely understand and respect that that's an important consideration. I mean, do we trust patients then? It's just, it's a matter, it's not that they're going to misbehave, it's just that they're going to behave based on, what they can afford to do. So interestingly, this exact idea is what one of the newer sort of what I would say forward-thinking laboratories is thinking about, is exactly that. Their sort of theory behind this is you get a lot of amount, huge amount potentially of information, which is going to come up with a whole lot of variants of uncertain significance, but being worried about this exact problem, about the providers continuing to work with that, they empower the patient, right? It's kind of the 23andMe type of thing, except it's not, just so you know, it's not 23andMe. But you empower with the patient, and they literally have a portal that you can take those variations. You don't even, you can just plug the nomenclature directly in there, and up will come. The information right now they're saying is just how frequently they've seen that. It's not quite as refined as we'd like it to be in terms of this is the new interpretation of what that means, or here are the health complications. But one theoretically could think of a system where you could do something right like this. So just so you guys know, from a medical point of view, from any of us that are here, there's a central resource called ClinVar, which is actually the aggregation of all of both the research enterprise, the clinical diagnostic enterprise, trying to pool, it's like crowdsourcing in terms of variant interpretation, all of them dumping their data as, it's de-identified, but with any particular variant being able to put their current classification of how they interpret that variant. And as I said, and these are very good laboratories. I'm not, taking anyone over the rails. Smart people disagree, and so you can actually see, you can pull up a variant and you can see smart people, smart laboratories disagreeing about the interpretation. But on the other hand, when you start seeing consensus in terms of everyone agreeing that this is clearly benign or this is clearly pathological, you start feeling more confident in the results that you're seeing. And over time, what will happen with ClinVar is that there will be, if you will, gene experts or subject matter experts that are very, very knowledgeable about particular genes that can really, at a higher level, curate that data and hopefully come up with correlations with functional data as well as other things. And so there are groups, for instance, two of the best studied genes on the face of the earth are BRCA1 and 2, and so there happen to be real gene gurus for BRCA1 and 2 that can be very helpful with that. So the reason I bring this up is because I do think it's a very interesting dilemma. I think your solution is actually a very potentially practical solution. I will say that in terms of the sort of traditional medical establishment, there are some people that get very uncomfortable with that idea. But on the other hand, I think you said it correctly. Who cares most about this information? It's the patient themselves who's most motivated to be able to, hopefully if they understand what it means to be motivated to follow this up, it's the patient. And so the question, I'm going to be provocative here, why not be able to empower the patient to do what's in their own best interest? So anyway, I put that out there because I think it's an interesting question to think about as we build health systems, how we might be able to do that, and especially as we go to an electronic health portal, how might you, for the informaticians, be able to build that into a system that you might have with that? Okay, so these are just very explicit now demonstrations in terms of that. Here's a silent variation, so in other words, a single nucleotide you might change, but it has no effect on the amino acids. Here's a missense variation, again, a single nucleotide that's changed, but now this changes an alanine to a proline. In this particular case, a single nucleotide that's also changed, but now this takes a lysine to a stop codon. So you can see, at the DNA level, don't look very much different, but have profound effects in terms of the proteins. And this, again, taking out a single nucleotide, leading to that frame shift and a premature termination. And again, these are just these splicing errors that we can sometimes see causing different gene mutations. And then in certain cases, this is for anyone who's a pulmonologist, the case of cystic fibrosis, where we actually now realize that sort of how severe these alleles can depend actually on intronic variations in the number of T's that are within the intron that may interact with the actual amino acid sequence. So it can actually be quite complicated. And I can tell you historically, we didn't appreciate how complicated this was as we started cystic fibrosis testing. So I'm just going to very briefly say this is a very fascinating form of hemophilia that actually gets better as little boys go through puberty, which is good for them, but that's because there happens to be mutation within the regulatory sequence, but just making the point that there can be mutations in those regions, and I'll just skip over that for quickness, and then this is an example of one of those triplet repeat disorders. So let me just briefly say then, and these are deletions. So let me just briefly say that this idea of variant interpretation, though, is quite common. And so for those of you who might be clinicians reading a report, I want to give you a few rules of thumb, and we'll actually come back to this when we're talking about how to read a genetic test. One of these is that we consider something that has an allele frequency, or in other words, a prevalence in the population of 1% or greater, to be something that generally we don't pay much attention to. We figure if 1% of people can go around and have that, it can't be too bad, at least when we're thinking about most severe diseases. So we oftentimes look at that as information. You should think, though, about diversity within communities. And so oftentimes the amount of information we have about those allele frequencies is really great if you're of Caucasian European ancestry. If you happen to be from a remote part of Africa, for instance, that might be very different. We might not have actually good allele frequency data from your particular part of the world, and so we might actually misinterpret information simply because we don't have good reference data. This will improve over time, but I just warn you about that. We tend to, and this may or may not be correct, but we tend to think that things that cause those premature terminations, those truncating variants, we almost always reflexively assume they're bad. That's not always right, so I'll warn you about that. In particular, when those tend to be at the ends of proteins, oftentimes they're not as bad as you might think they are. You can miss a little tiny bit of the end of your protein and still be okay. Splice variants are very tough. We have all sorts of prediction models that go with this, but they are just prediction models, and the same thing for missense variants. I will say, for those of you that are in terms of wanting to or interested in interpreting genomes, these algorithms for interpreting missense variants, I think, are getting remarkably better over time. They're actually using several different things, looking at cross-species conservation, looking at three-dimensional structures of proteins, looking at the type of amino acid you have, looking at the active site of the protein, all sorts of using all this type of information to come up with kind of one aggregate score, which I actually think is very, very helpful in terms of being able to just figure this out. Again, this is the type of thing that usually, on average, it's probably about 80% right. The problem and the concern for many people is it's not 100%. And so, you know, when are you going to bet the farm when you're far less than 100% on these things? Okay, so let's come back to our patient and let's figure out what happens here, because I know you're all anxious to hear the end of the story. So this is our little girl here who died, and she had these 3 genetic variations. She had, I don't know if you can see this well enough, I apologize, it's small, but she had a variant in CAV3, SCN5A, and the ryanodine receptor. And so the one thing that we decided, or I decided, I should say, was that we would start looking at the other members of her family. We would look at her sister, we would look at her parents, and see if we saw that they carried any of these variations. And in particular, these were all cardiac conditions. We'd also look very carefully at their heart. We'd look at their EKGs, we'd look at their echocardiograms, and see if we could start seeing any correlation between the genetic findings and what we were seeing in the parents. Can you guys see this well enough? Okay. Does anyone, if anyone want to take a hazard, if you had to pick one of these 3 variations, CAV3, SCN5A, the ryanodine receptor 2, does anyone want to make a guess about what I was most concerned about at the end of the day? So what this means, just to give you the nomenclature, positive means they actually have the genetic variants, the rare variants. Minus means they don't have it. They just have the normal sequence at that position. And so for those of you who can't read it because it's too small, I'll tell you that Dad has none of those 3 variants, Mom has the top 2 of the variants, and her little sister has none of the variants. And within all of these, I'll also tell you the gobbledygook that's on here is those EKGs are normal. So of those 3 variants, the question is, of those 3 variants, which one are you worried about based on looking at the family members? The ryanodine receptor. You're absolutely right. You've learned something. This is fantastic. So why are you worried about the ryanodine receptor? That is absolutely the right answer. Because she's the only one that has it, right? So in this particular case, and I will tell you, because some of you might be worried, Dad doesn't have any of these 3 variants. He actually is her father biologically, so don't worry that there's a milkman involved in this. He really is her father biologically. But he doesn't have any of these. Her little sister didn't have any of these, thankfully. That made it a lot easier for the family. And actually what we know looking from the mom is that 2 of these are inherited, but the ryanodine receptor is actually de novo in the little girl. So it started brand new with her. And that particular condition is associated with catecholaminergic polymorphic ventricular tachycardia. So now let me tell you a little bit about that condition. As those clinicians can think about it, dissecting the words, what happens in those cases is that any individual, if they get, you know, this big adrenaline rush, if they get, you know, excited or scared or anything like that, they go into a bad arrhythmia is the bottom line on this. And it can be so bad that, in fact, your heart could stop, or it could beat apparently that it's not effective. And so in each of the cases, remember when her cousin pushed her into the pool, she basically got freaked out because she didn't know how to swim. And so that caused this adrenaline rush, that catecholamine. Her heart briefly went into one of these bad rhythms. It actually spontaneously went out. Of course, I'm making this up. I assume this is what happens as a hindsight. And then the other circumstance, I actually think, again, she got scared when she was in the water, not being a confident swimmer. Unfortunately, this happened again, but in both of those cases going under the water, but being more catastrophic the second time, of course. And so, in fact, with this information, interestingly enough, I've been following her mom over time because we still had these two variants that still are not totally in the clear with mom, but mom's been perfectly fine. There's no family history of anything in mom. My suspicion is that those other two variants absolutely mean nothing, but that her, in fact, definitive diagnosis was CPVT due to now an identified mutation and being able to take the other family members largely off the hook, but especially her little sister off the hook from doing this. So you guys already answered this question. You figured out that what happened in her particular case. And again, as I said, you came up with the right conclusion that this is likely, this is likely the major risk factor for her, and thankfully the only one in the family. This, although I don't know if those of you who are parents can put yourself in that situation, I can tell you from an emotional point of view, this was certainly, it didn't bring their daughter back to life, but actually gave them a lot of information and a lot of closure about what happened. Otherwise, they were going back and forth in terms of trying to figure out what happened, and most importantly, as I said, about their other little girl, whether she was going to be safe. And so I think even though this is something that clearly didn't bring their daughter back, I think having that diagnosis actually was extremely helpful for them. So just to wrap up and in summary, the good news about this is all getting better, but the point that I want to make is we are very, very much still in flux in terms of how to interpret an exome, a genome, even one individual gene. And so I think the greatest challenge for us is to be able to do it as best we can, and to a certain extent that comes with how we start talking about uncertainty, how we talk to patients about uncertainty, how we correctly convey how certain or uncertain we are about certain bits of information. And for some people, some patients, that can be a very sophisticated sort of conversation. So we've got to figure out a way of being able to do that better. We've got to be able to find a way of helping individuals live with ambiguity for some certain period of time until we resolve that ambiguity, which I am absolutely confident will happen over time. And then to me, most importantly, we've got to figure out how to have this long-term relationship with our patients, or at least they can have a long-term relationship with their data so that they can ultimately come up with the best information for themselves in terms of taking care of themselves and their families.